the very first thing that struck me, I went to a supply chain discussion first thing this morning, and the thing that really struck me was the hatred between academia and small companies and big pharma. And it was so funny, because it was like, you know, all these academics going, well, academic GMP facilities going, well, we've got these supply chain issues and we can't get our products, but big pharma have got so much more buying power and they can do it all. And then finally somebody from big pharma stood up and went, I'm from big pharma, I just want to say we have the same problems. But supply chain issues are still an issue for everybody. And I think what they were saying, which I think is where we go with this, is don't go with something at the beginning. When you're first developing your process, look at alternatives, because if you can't get one filter, you're going to have to put another one in. So I think that's the take home. The supply chain is not going to go away anytime soon. We've got, this industry is just growing exponentially, and suppliers can't keep up. It's kind of inevitable. So, be pragmatic in your process development. Don't pick something that you've only got one supplier for, and that's it. So that was one. And then the other thing, which is a bit disappointing, I think, although personally from an eXmoor perspective, it's an opportunity. All the academics for me were still in the same place that they were in. Not all the academics. A number of academics are still in the same place they were in 10 years ago, which is they really still don't understand how you get your product from research to manufacturing. And there's a lot of confusion about terminology, and people are saying GMP grade reagents, and then somebody else rightly said there's no such thing, and then someone else has talked about pre-GMP, and some guy was going, what does pre-GMP mean? How do you define that? And it's, well, actually it doesn't mean anything, there's no definition. So there's still a lot of confusion. And academics and early spin-outs still need a lot of guidance. From an eXmoor perspective, that's fab, because we can help them with our CMC strategies early on. But if we're going to be successful, we've got to kind of get that education right the way down from people who are big pharma all the way back to academics. So we're well on our way now to becoming a fully fledged CDMO. We announced our funding yesterday, which we achieved amazingly in the current climate in less than 6 months. So I'm very excited that we managed to do that. And we move into our new manufacturing facilities in July, which is very close. Literally the first week in July we move. We've handed in our notice on our current facilities, which are in an incubator center north of Bristol, and our manufacturing facility is also north of Bristol, but it's on a purpose-built site. So that's one big change. But I think we're becoming a CDMO with a twist, I guess, because we're building on that kind of 15 plus years of consulting experience with CMC strategies and helping people design their facilities. And then we're building on 5 years of work in our PD, our process development and analytical development labs. And we're very focused on getting the CMC strategy right, the process development right, with the confidence that the manufacturing will kind of drop off the end. So whereas most CDMOs are really focused on manufacturing and selling manufacturing slots, we're interested in making sure that they've got the right process to go into manufacturing, and that they're following the right CMC strategy, which will get them, hopefully accelerate them to the clinic, as opposed to get them to phase 1 and then they can kind of circle around a few times and then maybe move on. And our PD team is 30 odd people now, and that continues to grow. So we don't look like your traditional CDMO, but that's what we are being labelled, and that's what people understand. But we have a lot more capability to offer than just manufacturing, and I think that's going to be critical as we move forward in this industry. Everybody's putting up facilities. There's lots of capacity coming. It's not the same as capability, and that's I think what's going to be our USP, if you like. We're trying to accelerate people's products to the clinic because that's the business problem they're trying to solve, and also it's the problem the patients want to see the solution to. So yeah, that's where we are. And compared to when we talked last time, we had just, I think we were 18 months into our PD lab, so it was still quite new. And now we're 5 years down the road. So in the labs, when I talked to you last time, we were just doing viral vectors. Now we do viral vectors and allo cell therapies, MSCs and iPSCs predominantly. And now this year we're just moving into autologous cell therapies in the lab, and then obviously we're moving into GMP. Funnily enough, the pandemic was almost incidental from an eXmoor perspective. It kind of didn't change anything. We had a plan, we were working on it. It didn't disrupt the work. I mean, when it first started, I was thinking, oh no, everyone's going to cancel their projects, and actually nobody did. And so, we just kind of motored on, and it was kind of incidental, obviously not from a personal perspective, but from a business perspective, it was. I think the other thing that's changed in the interim is, in order to get ready for this big expansion, I've kind of formed a really strong management team, which I didn't have before. So 2019, it was me and really my other half, to be honest, that were kind of doing most of the company leadership. And now we've got a very strong senior leadership team, and now a new board with our new investor coming in. So it's a very different company. We've got a CFO, and wow, what a revolution that was, so like, I don't have to do the spreadsheets myself. One that comes to mind that's big in the conference is iPSCs. So they're so much, iPSC derived products are so much more complicated, because before we can do any process development, you have to have acquired a cell line, and I'm talking allo now, allo iPSC. So you have to have acquired a cell line. It has to be appropriate. It has to have freedom to operate around it. It has to have kind of a track record. And that's not easy to get in itself. But having got that, you then need to bank it. You then want to gene edit it. You want to bank it again, probably. And the whole set of regulations and guidance around that is very, very confusing. Where do you start GMP? And if you start it right back at the beginning, then it means that your first cell bank and then your gene editing and your second cell bank are incredibly expensive to build, all before you've got any phase 1 data that says your product's any good. So there's a decision around there of do you do this knowing that you probably need it in the end, or do you say, well, actually, let me get to phase 1 in the full knowledge that I might have to go all the way back and do it all again to kind of move on. And the reason that's important to us is because we can't start process development until they've done they've got those things in place. And we've got at least one client where we had hoped to start 6 months ago, but that whole thing has caused a big delay to them, and so they can't start, we can't start. So that's one area. I think the other area is partnering in a more true sense of the word with your service provider. So as a CDMO, not expecting them to just lob something over the fence and we lob it back when it's done, but much more taking the time to think about, well, what is your expertise? What is our expertise? We shouldn't do the things which you're better at. Let's work out more of a hybrid way of working rather than a straight kind of cut and dry traditional method. And I think that's going to be important for these more complex products. Viral vectors may be fine because that's actually, in the scheme of things, relatively straightforward. But you come to an iPSC and you're thinking, well, there's a particular kind of assay that you've developed in-house. You're really good at it. Why would we take that off you? Why don't you carry on doing that? And we'll do the bioreactor package because we're really good at that. So I think that and also there's definitely, I've seen a desire to work more closely together so that at the end of the day, it's their product and their process, and they want to take it. They might want to take it somewhere else. They might want us to build them their own facility, and that's absolutely fine. It's what's right for their business model. The whole acquiring of the cell line in the beginning is important, and I don't know how we get to a place where there's some really good providers, that people can trust and can get their cells from. But that's certainly one of the big issues. The other one, as I described, is the level of GMP you use in your cell banking and your cell line development. But then I think moving on from there, every iPSC program we've had in the lab has come to us in a 6-well plate, and clearly a 6-well plate is not going to make it to commercial. Which is not, when I go back to when we started in cell therapies in 2007, our first project was regulatory T cells in 6-well plates, and so it's kind of that, that product has moved massively in the right direction, and this will too, but it's a little bit more tricky, obviously, because now these are adherent cells, so now we're talking about microcarriers in a bioreactor, but all of that is very feasible, I think, and will be a set of challenges that we can overcome. And it's kind of keeping, because if these are allogeneic products, so there's a potential mass market. So we do need to seriously think about scale and cost of goods, and and so on and so on. So thinking about those things early in PD is, I think, is one of the challenges. People have said, since I started, oh, well, autologous therapies aren't going to last, it'll all be allo. And now people are saying, oh, viral vectors are not going to last, it's going to be non-viral deliveries or whatever. My personal feeling is that there's a space for everything, and it'll be indication dependent. I think the biggest opportunity is to move into mass markets. So instead of solving the monogenetic diseases or the orphan drugs, it's people who are now thinking about solving the big diseases, the liver disease, Parkinson's, dementia, heart disease, all the things which are massive. And the way to solve those is really getting your processes robust, whether it's a scale out or a scale up. It's really focusing on getting that process development right, and and that's going to be where cell and gene therapy finally really makes a big time, because these are millions of patients, not hundreds of patients, and those diseases really need treatments.